Hello everybody, Namaskar. In this lecture today, we will discuss about the smart packaging. Okay. The concept that we will cover include smart packaging, active packaging systems like active scavenging and absorbents, active emitters, antimicrobial agents, or intelligent packaging like sensors, time temperature indicators, microbial indicators, O2 CO2 indicators, and finally, we will take up some case study. Okay. So, let us see what smart packaging means. Means it is an advanced packaging technology which offers additional functions apart from the traditional packaging. In the earlier lectures, we have discussed about the packaging technology. Okay, but the smart packaging has something additional to offer. The smart packaging offers benefits such as it prevents the microbial growth and oxidation of food. It indicates the on-time quality of the food. Smart packaging senses and informs related to the internal or external conditions change, that is what is the condition inside the package, and it also indicates seal integrity and act against counter faulting or if there is any leakage etc. or not. So, it communicates additional food information to the user and respond to food condition changes or packaging environment. So, basically this smart package system, they work upon the interactions between the food inside, the environment inside the food package and the packaging material characteristics, and they sometimes inform to the consumer the condition of the food inside, or they regulate the environment and depending upon their interaction and extend the shelf life of the food. Okay. So, the smart packaging systems can be classified into two categories. One is the active packaging that extends the shelf life based upon the interaction between the food characteristics, environment inside the package and the packaging characteristics. And the intelligent packaging system, it communicates with the consumer, that tells yes, the food inside it is not good, it is good, its quality is to that it is fresh or not. So, it provides or provide the information about the material inside the package. So, let us see first active packaging. As I told you, active packaging is a type of packaging where certain components are incorporated into the packaging system that releases or absorbs substances from or into the packaged food or surrounding environment to prolong the shelf life of the food and to sustain the quality, safety and sensory characteristics of the food. It can be described as a technology devoted to controlling the rate of respiration or the growth of microbial, delayed oxidation, moisture migration with the main purpose to enhance the food quality, shelf life, freshness, safety and so on. So, the active packaging basic concept is, I told you, integrating specific substances in the food packaging in order to control or extend the quality and enhance the shelf life. The active packaging system, many kinds of natural and synthetic active compounds like plant extracts, essential oils, peptides, enzymes, organic acids, metal ions, metal oxides, nanoparticles etc. are used in the packaging material which impart different functionality, such as releasing or emitting of antioxidants, antimicrobial, sulfur dioxide, preservatives, ethanols, flavors and so on. It may also act as absorber or scavenging of carbon dioxide, oxygen, ethylene, flavors, moisture, UV light, so on, or it acts as a blocking or barrier agent, or it controls the microbial growth or temperature and quality of the food. So, the routes for obtaining the active packaging system, that is like A may be incorporation of external devices, that the labels, pads or sachets loaded with some active compounds, that polyvac or other things they can be taken, and here either emitter or absorber, they in the calculated amount, and the quantity of these absorbents or emitters is calculated based upon the known characteristic, that is respiratory behavior, transpiration and the permeability of the packaging materials, etc., permeability to oxygen, as we discussed in the earlier class. So, all these things data can be used to calculate, that is an extended shelf life, intended shelf life, how much shelf life one wants, whether you want to hasten the respiration or you want to delay the respiration or any particular change. Accordingly, that amount of the absorbent or emitters can be calculated and they can be inserted into the package. Alternatively, that is these absorber emitter or these materials, they can be coated, that is the coating of the active compound into inner surface of packaging material, like antimicrobial substances, etc. They are interior of the packaging material, they are coated. So, this comes in contact with the surface of the food material during when it is packaged, and then surface microorganisms are inactivated or killed and the shelf life of the food is extended. Okay. This system is useful for heat sensitive components are incompatible and immiscible with the polymeric matrix. Then there may be immobilization of active compound in the inner layer of the packaging material through either ion or covalent linkages, and it is important to have the presence of functional groups on active compounds and polymer to get immobilized. That is, the functional group for the immobilization should be there on the packaging surface. So, also there may be alternatively, like in the case of D, you can see here that is the direct addition of the active compound into the packaging material matrix, and the packaging material is being made, okay, like this some antioxidants or some other functional components, they can be added, incorporated into the packaging material. So, this becomes functional packaging or even materials or antioxidant rich packaging material, etc. And this when used for packaging, it extends the desirable properties, or by using polymers with some active functions like chitosan to develop the compounds or multi-layer materials for use in the pack. So, these are the different ways by which we can get the desired environment and desired quality, etc. of the food, or we can do the active packaging. So, basically the different active packaging agents, that they are used with the different roles in the active packaging of fruits and vegetables, and these major roles are that is like their oxygen scavenger, they are used to as a oxygen scavengers or carbon dioxide scavengers or ethylene scavengers or emitters, as the case may be, antimicrobial releasers or antioxidant releases, etc. Okay. Then we will let us have little details of one by one these things, like oxygen scavengers, that is it could be applied effectively to reduce the food quality loss due to oxidation of sensitive food or prevent microorganism growth due to the presence of oxygen. Okay. Also, these oxygen scavengers in the control atmosphere storage or in the modified atmosphere storage, where we want that is to extend the shelf life of fruits and vegetables, their oxygen scavengers are, because we have to keep the oxygen level at minimum required level to continue to allow the respiration, aerobic respiration at a slow rate as possible. So, their calculated amount of oxygen scavengers are added, okay, or carbon dioxide scavengers also for that purpose. Carbon dioxide scavengers are employed to removal of the carbon dioxide, okay, because when the fruits and vegetables they respire, they release carbon dioxide. So, we want that inside the package a particular concentration of carbon dioxide should be maintained. So, the scavengers can be added into the packaging system, active packaging system, to absorb the oxygen, that is carbon dioxide. So, these are the result. Okay. So, various the CO oxygen scavengers chemicals which are iron, ascorbic acid, enzymes, salts, fatty acids, etc., or linoleic acid, or photosensitive dye, or some of the carbon dioxide. Known carbon dioxide scavengers include calcium hydroxide, which reacts with carbon dioxide at a humidity to create calcium carbonate inside the food packaging, etc. Similarly, the ethylene scavengers or ethylene emitters, as the case may be, that ethylene causes the fruit and vegetable yellowing. It is a ripening hormone. So, it leads to result spot and has shelf life reduction effect of many fruits and vegetables. So, potassium permanganate is used to scavenge ethylene, okay, that oxidizes ethylene to produce ethanol and acetate. Okay, thus reduces the ethylene content inside the package, and you, by reducing the ethylene content, one is reducing the ripening rate, okay, and the shelf life of material is extended. Similarly, if you want to enhance the ripening of the fruit, then inside the packet some ethylene releasers can be used. Similarly, there are the odor and flavor absorbers can be used to remove undesirable flavor substances produced due to various oxidative and biochemical processes occurring in the food, or by the removal of such undesirable flavor components, acceptability of the fresh foods can be improved. This moisture absorbers, very commonly used, that is absorbers in the packaging materials. That moisture control is required to prevent condensation when fresh horticultural produces respire, and this is very important in the case of equilibrium humidity packaging, EHP. So, the condensed moisture, if the equilibrium humidity inside the package and outside the package is not uniform, then there may be condensation of moisture beneath the packet, and it may encourage a microbial growth, fungal attacks, etc. So, the moisture control inside the package is very important, and it should be done precisely. It should be precisely regulated. Excessive moisture loss can lead to shrinkage and toughening of the texture. Desiccants are being successfully used for a wide range of foods such as chips, nuts, candies, gums, spices, fruits, vegetables, etc. The carbon dioxide emitter, that is it, carbon dioxide, it has a direct antimicrobial effect. It increases lag phase and the time during logarithmic phase. So, that sometimes to the raised level of CO2 is maintained inside the inside the package to control the aerobic bacteria or aerobic respiration, etc. So, the carbon dioxide emitters are used. Carbon dioxide is produced inside the package by the food reaction with sodium carbonate and citric acid mixtures inside the dip pad. Ethanol emitters, etc. Accordingly, ethanol emitters are extensively employed due to the capability to retard or slow down the process of mold growth in the food products. The sachets comprising that 55% ethanol and 10% vapor, which were absorbed into the 35% SiO2 powder, then filled into a sachet of a paper ethylene vinyl acetyl copolymer, were used for antimicrobial effect on food packaging. Then similarly, various antioxidant releasers, that is, they are integrated into the packaging or coated with the packaging material surface to control oxidation of the fatty components and pigments, etc. Even plastic films like polyolefins are impregnated with antioxidant for making a stable polymer and protect against oxidation. Okay. And antioxidative activity could also be found in plant extract and spice extract that consist of polyphenol, etc., and they can also be impregnated with the packaging material. Then sulfur dioxide emitters work based on the metabisulfite hydrolysis mechanism and the reaction of calcium sulfide with moisture. So, sulfur dioxide emitters has been used for grape packaging to prevent mold developments. A polymer has to be applied for sulfur dioxide control release to obtain inactivation of fungal without the undesirable effect on the food. So, similarly, now there are various antimicrobial agents also, like niacin is a bacteriocin in the plant extract, that allicin, thiosulfonates, proteins are chelating agents like EDTA, enzymes, peroxidase, enzymes, fungicides. So, there are various antimicrobial agents, and these antimicrobial activity in active packaging can be classified as blocking or barrier functions that prevent microbial growth in food packaging. Antimicrobial food packaging can be classed into 4 antimicrobial packaging types, that is where antimicrobial agents are impregnated into a sachet and attached to the packet, or antimicrobials absorbed or coated into the surface of the polymer. Antimicrobial agents are integrated into the packaging film directly, or packaging materials have a matrix that consists of an antimicrobial agent. So, this is either of these depending upon the type of the material, type of the food which is being packaged, interaction, these antimicrobials can be integrated with the packaging system. So, these were the different ways of that active packaging systems. Now, let us discuss about the intelligent packaging. As I told you earlier, that intelligent packaging continuously monitors food quality during distribution, storage and selling, or even to the end customer, and it informs the customer about the quality and other parameters of the food inside. So, in the intelligent packaging, various sensors are used. They work on the principle of detection of biological and chemical compounds like enzyme, DNA, etc., or detection of chemical compounds like reagent, dye, pH indicators, etc., or there are freshness indicators which work on the principle of detecting biological and chemical compounds, ethylene gas detection, pH detection, or integrity detectors which that is the oxygen and CO2 indicators, leak indicators, etc., time temperature indicators which work on the diffusion, or enzymatic, microbial, photochemical sense, or RFID, radio frequency technology is used for the tagging of the material. RFID tags are used to trace the product during its communication when it is being transported, shipping, etc. So, the various these devices are used in the intelligent packaging system. Okay. The sensors, first let us discuss. A sensor can be defined as a device or tool used to quantify energy of matter as a signal for a chemical or physical property detection or measurement to which it responds. So, various components of a sensor include a receptor, that is are the sensing part of a sensor, and this is the part which comes into contact with the analyte and produces a physicochemical signal. So, there is other component which is transducer element, and this transforms the physicochemical signal from the receptor into a useful analytic signal. Then this signal processing unit, it is sent, this analytic signal is sent to the signal processing unit by the transducer, and this process, now this signal processing unit is process the output of the transducer to get the desired information, desired output form, and finally which is sent to the display unit, where quantifiable results are displayed either in analog or digital form. Okay. That is how this. So, various type of sensors may be either chemical sensors, gas sensors, or biosensors, and they all have the transducer, as I told you, either optical, electrochemical, or electrical devices like metal oxide, semiconductors, etc., or thermomercury, magnetic devices, etc. So, these are the various components of the sensors. Then time temperature indicators, TTI level. TTI are a small and inexpensive device attached to a packaging material that provides time temperature history of the food product during storage, thus estimating the remained shelf life in the non-destructive manner. Okay. Like here you see that it is there are time temperature labels depending upon the exposure times. That is the various enzymatic reactions are indicators are the pH, how if the pH by as the time progresses the pH changes, and the change in the pH that is may indicate that is these indicator, it will change the color. So, it may be pH indicator, it may be a microbial load indicator, moisture indicator, and it can be designed to have at a particular content of the particular quality, the color of the sensor TTI level will change. So, these TTI level indicator, TTI indicators, they function based on that, they can be classified based on their function, like critical temperature indicators, critical temperature time indicators, time temperature indicators are they may be based on the operation principle like mechanical, enzymatic, chemicals, microbiological, etc., or they may be based upon the history of the this temperature like partial history indicators or full history indicators, that is the package, etc. So, various types of TTI indicators are used in the intelligent packaging. Then microbiological indicators, this is the information on the food quality is usually based on chemical changes on microbial growth within the food. Microbiological quality is indicated by the reaction of the indicator with the microbial metabolites. That is food quality. There may be indirect indicators like the critical temperature indicators, time temperature indicators, leak indicators, that is the it may that indicators may say that it may show that there is a leakage. So, obviously, when there is a leakage, the inside the gaseous composition will not be properly maintained. So, the shelf life will be adversely affected. So, these are the indirect indicators. The direct indicators may be like pathogenic bacteria, count of a pathogenic bacteria, their number can be indicated by these TTI indicators, or even biogenic amines, ATP degradation, aroma compounds, etc. So, various volatile compounds, CO2, nitrogen. So, these their values label inside the package can be directly indicated, by which you know that once the label goes beyond a certain limit, we can say that yes, the food is good or bad. Okay. Then O2 and CO2 indicators, the shelf life of the fruits and vegetable, as you know, depends upon the rate of respiration that can be expressed as oxygen consumed or CO2 evolved, which further causes changes in the food color. So, when the respiration rate is increased, the production of ethylene is increased. These properties can be implied as marker or indicator for the freshness and the quality, okay, the food. So, the indicators must be directly contact with the gaseous environment immediately surrounding the food in a container, and the presence of the gas may indicate that the package was sealed incorrectly after leaking or damaged. So, the plastic optical fluorescent films are highly sensitive for the detection of the gases and dissolved carbon dioxide, etc. So, these leak indicators, etc., they you can various indicators, they can change in the color, etc. They can show that yes, there is a package leakage. Then smart packaging systems for fruits and vegetable freshness allows continuous monitoring of the quality of packaged food, fruit and vegetable. Fruit freshness sensors are on package indicators that monitor environment inside the outside the packet. It allows consumers to make informed decision about the quality of the fruit. So, the sensor it's the signals like in the food packaging systems, okay, but one has to need to calibrate the signal, and then this sensor, which are the signals sensor inputs finally are released, as I told you earlier that it is released in the output in the color, fading, voltage are in various forms, and from that one can infer that as quality of the food inside. So, we will take a few case studies like some active packaging of fruits and vegetables, where action of self generating chlorine dioxide Cl2 gas for fruits and vegetables packaging systems containing moisture activated chlorine dioxide gas releasing label. You can see here in the figure okay, CO2 gas lowers the metabolism of fresh produce, which help in maintaining weight and firmness of the fresh produce during their storage period. So, CO2 gas prevents fruits from softening associated with its capability to prevent protein synthesis in cell walls. Compared to the control sample, a lower weight loss and a lower firmness value were noticed in tomatoes packaged in chlorine dioxide gas. So, here you see these are the NaCl2 indicators, acids, threats and sources of ClO2. So, NaClO2 plus acid plus moisture, and all these three gives ClO2 gas. So, this is the ClO2 releasing label. Okay, ClO2 gas comes and this automatic release of chlorine dioxide, but it extends the shelf life. They are put outside in the packaging material. So, this the quality of the tomato or other fruit is accordingly increased. Here it is the freshness indicators of the apple. You can see that is this red line is the degree of sensory ripeness, and the black line shows the ethylene concentration in the. So, a relationship between ethylene concentration and degree of sensory ripeness is shown here, and it is with the changes with the storage time in days. Okay. So, ethylene emission of apples can be detected through the ripeness indicators. This indicator is based on this are the publication by Lang and Hubbard. So, this indicator is based on the reduction effect of ethylene causing color change in selected metal ions. The used molybdenum chromophores change under impact of ethylene in color spectrum from white light yellow to blue because of partial reduction in molybdenum MO to MOV. So, the color changes from white to blue in the ripeness indicator labels on the apples were clearly visible over the studied time. Okay. So, this you can find out this. Then another this application of freshness indicators of apple, where that even zinc oxide, that is nano zinc oxide powder. So, a novel packaging nano packaging was synthesized to study by coating the polyvinyl chloride film with nano zinc powder, nano zinc oxide powder. Here you can see. So, on investigating its effect on preservation quality of fresh cut Fuji apple, and nano packaging significantly reduced the fruit decay rate. Both the polyphenol oxidase and pyrogallyl peroxidase activities were decreased in nano packaging fruits. The initial appearance of apple slices were retained and the browning index was preserved in nano packaging samples, and these results indicated that nano zinc oxide active packaging could be a viable alternative to common technologies for improving the shelf life properties of Fuji apples as a fresh cut products. So, there are various such similar examples where these even they using the nano science and nanotechnology to nano sensors etc. are applied, and they can be used both in the consumer packs or in the large scale retail packs etc. to tell the information about. Even there are certain self heating and sealing pouches are available now, packaging materials. They are that is when you open the packet and it may give that desired water absorption effect etc., instant oxidation reaction. So, there are various types of packaging, that is packaging material and active packaging or intelligent packaging. So, finally, I will summarize that yes, smart packaging is a combination of both active and intelligent packaging system that integrate the shelf life of the food. These monitor the shelf life, they extend the shelf life of the food, and they also monitor the quality, and they improve the quality as well as inform the condition of the food inside in terms of its particular quality, maybe color, flavor, microbiological value, or in terms of its shelf life, it may say that yes, it is spoiled or it is fresh. So, any system etc., can be used. Okay. So, to indicate that that is the role of the smart packaging. So, in the active packaging, various absorbers, emitters or barriers and control system are used, whereas in the intelligent packaging, there is various indicators or sensors etc., are used to measure and inform about the quality of the food. So, this are the references used in this lecture, and this, thank you very much. Okay, na?